Okay, should we go ahead and get started? Ready? Okay. Good morning or good evening in case of our first speaker. Welcome to the second day of the symposium. We want to thank everybody for being here. And just as a word of business, I guess, today happens to be World Kidney Day. And they are celebrating that by socking it to the kidney with interesting socks. So if you have a interesting pair of socks on, Terry, I didn't tell her this beforehand, is going to accept all those. And we're going to put them all up on our Blue Sky link if we can. So please participate in that wonderful time to be in kidney research. Let's all work together to solve or slow the pace of kidney disease. And so just as a kind of reminder of the PKD RRC, and our mission is to develop and share innovative resources, reagents, and our expertise to advance, innovative research into the fields of polycystic kidney disease. Today we have an interesting lineup. We're going to start out with therapeutic targets in PKD, move on to non-PKD1 or PKD2 forms of renal cystic diseases. We'll have a short break for lunch. We'll come back and spend some time not talking about kidney, but the other manifestations of PKD and other tissues and other model organisms to study the pathways associated with PKD. And finally, if you're a trainee, we would love to have you join us at our trainee workshop at 3:15. Again, this is Eastern time. Separate link has been sent for that. If you need more information, Terry or Nicole would be happy to have you, send an email to them, and they're happy to get you involved and get you registered for that event. So kind of housekeeping as similar as yesterday. Everybody will be muted. No cameras will be on except for the speakers, the session chairs, or panelists if they're there. We are really on a tight schedule, so we're going to try to keep everybody to time. We will give speakers a 5-minute warning. And we're going to handle all of our questions through the chat functions where the moderators will ask the questions for you. You can enter those questions at any time you want to, and then they will be followed up for. If we can't get to them during the session, we'll follow up on them offline or after the speaker is done. Also, we will distribute a survey a couple days after the symposium. Please fill that out. We use that information to make adjustments for next year's symposium to make it better and more useful for those that participate. We would love to have you guys visit our site. You'll find a lot of information up there with regards to resources, new projects that are coming out that might be of use for you. We also announce our pilot projects there. Updates on the annual symposium, which again is today, but for the next one, as well as a monthly scientific roundtable, which have been really well attended and really great talks to interact with fellow PKD researchers. We would love to have you guys join us on Blue Sky. Follow us if you're a member of Blue Sky. Sign up for our mailing list and the discussion boards. Also, since doing PKD research and communicating your ideas with colleagues is always an exciting thing to do. I want to remind everybody of the PKD FASTSIM meeting coming up in June 29th through July 31st in Southbridge, Massachusetts. This would not be possible without support from NIDDK, so thank you very much for helping us. So it's supporting the centers and our activities. A major thank you goes out to Patricia and Pam for being the chairs and organizing this whole symposium, but also the extensive amount of work that the committee put in to organize this. So a big thank you to all of them. And with that, I think we are done, and we're going to turn it over to our moderators who will introduce the first speaker.
Thank you, Brad. Good morning, all, and welcome to the first session on therapeutic targets in PKD. I'm Robin Mazer, along with my co-chair, Duameni Neamu, both of us from the University of Kansas Medical Center. And I'm very pleased to introduce our first speaker. This is Dr. Shuan Xia from the Nanyang Technological University. Dr. Xia earned her PhD in molecular and cell biology from the National University of Singapore in 2010. She pursued postdoctoral training in Dr. Juan Carlos Izpizua Belmonte's lab at the Salk Institute from 2010 until 2015. Since 2015, Dr. Xia has been a faculty member at the Li Kong Chian School of Medicine, Nanyang Technological University in Singapore. Her research focuses on creating human stem cell-based models to study kidney development and disease. Her talk this morning is entitled Reconstructing Tissue Microenvironments to Better Emulate Polycystic Kidney Disease. Dr. Xia.
Thank you, Robin, for the very kind introduction. Let me share my screen. Okay, I need to get it full screen. Okay, here we go. First, I would like to thank the meeting organizer for inviting me to give this presentation for this very exciting symposium, and also for very kindly arranging my talk in the first of the second day because of the time difference. My lab is essentially a stem cell biology lab. We have a long-standing interest in developing organoid models that can recapitulate some key structural and functional properties of the human kidney. We hope these models could complement the conventional animal models for studying pathophysiology that might be unique to the human kidney. So time really flies. It has been more than a decade since the concept of organoid has made its official debut in 2013 when several organoid and liver organ bud was successfully established from human pluripotent stem cells, manifesting tissue structure of higher sophistication and compartmentalization. The past decade has witnessed the explosion of organoid biology. Currently, a large variety of organoids can be generated in a dish using different types of source cells, human pluripotent stem cell, tissue-specific stem cell, or tumor cells. So these organoids have been very widely used for a wide range of research purposes, especially for disease modeling and drug testing. So coming back to our kidney, which is a vital metabolic organ regulating body fluid homeostasis, waste excretion, hormone production, adult human kidney is comprised of more than 20 different types of cells. Importantly, both the composition of the cells and also the structural organization of the cells are essential for realizing its full functional competence. So how can we recreate such a complex organ or even part of it in a cell culture dish? These are three landmark studies laying the foundation for the kidney organoid research field. The study by Dr. Melissa Little Group and Dr. Joe Bonmentry Group demonstrated for the first time it's feasible to differentiate human pluripotent stem cells into kidney organoids containing segmentally patterned nephron-like structures. Whereas the existence of ureteric structure in kidney organoids had been a little bit debated. So to this point, Dr. Nishi Nakamura group from Japan separately differentiated human pluripotent stem cells into ureteric bud structure, which can reconstruct higher order embryonic kidney-like tissue alongside pluripotent stem cell derived metanephric mesenchyme cells and mouse embryonic kidney-derived stroma progenitors. So currently, the most prevailing kidney organoid platform can be categorized into two different types depending on the embryonic origin. Probably I should try to smaller this. Sorry, I wondered how can I. Sorry, okay. Oh, it's better. Okay, so nephron organoids, or more often called kidney organoids, contain the main components of the nephron, from glomerulus to proximal tubule all the way to distal tubule. Whereas the ureteric organoids resemble the developing collecting duct with very well established tip trunk polarity. Today, my presentation will be mainly focused on the nephron organoid, or more generally called kidney organoid, because so far as my lab myself is concerned, we haven't been very successful in maturing the ureterical organoid to adopt a very good similarity to collecting duct-like structures. So this is a very representative image of human pluripotent stem cell-derived kidney organoids, which contain segmentally patterned nephron-like structures. They are adopting a sort of proximal to distal axis from glomerulus to proximal and all the way to middle segment, JAG1, and all the way to the distal tubule-like segment. And more importantly, these organoids contain a pretty elaborated vasculature network, although they still lack the proper patency reminiscent of the in vivo renal vasculature structure. To promote the maturation of these kidney organoids, the methodology we adopted is to engraft them into immunocompromised NSG mice under the subrenal capsule space. 4 weeks later, we recovered the graft. These kidney organoids were successfully anastomosed with the host circulatory system. As you can see, the red blood cells swimming within the glomerulus, and the Bowman capsule have been very nicely formed, and even tubule structures get slightly elongated. And more importantly, we observed the endothelial cells of human origin migrating or invading into the nearby glomerulus to establish the putative glomerular capillary tuft. At the ultrastructure level, podocyte formed a very elaborated food process, endothelial cells got fenestrated, even establishing a sort of putative glomerular filtration barrier alongside the basement membrane and the cellular process of the podocytes. When we inject the dextron of different molecular weight from the tail through the tail vein of the host animal, we injected the lower molecular weight inert dextron 70 kD and 2000 kD. 2 hours later, we recover the graft. We actually can detect low molecular weight dextron within the proximal tubules, but not the high molecular weight dextron, which appear to be stuck in the blood vessels. We speculated it is likely these dextron may be first filtered by the glomerulus-like structure and then reabsorbed by the proximal tubules. Sorry about the molecular weight, because actually we try to inject the same dextron of 70 kD into the mouse. They also can get filtered and reabsorbed. I guess probably it doesn't have a negative charge. So considering the limited physiological function manifested by these kidney organoids, what kind of kidney disease the current kidney organoids are best suitable to emulate? So my lab has adopted some criteria when we try to choose the disease to work with. We hope it's genetic kidney disease with characteristic structural abnormality. And also disease phenotype or manifestation does not rely on an extrarenal organ system. And third, we hope the disease phenotype can be visible to read without systemic biology approaches such as single cell all the time. So considering all these factors, polycystic kidney disease appears to be a very best pick due to its relatively clear genetic underpinning and visually distinct disease phenotype. So PKD can be broadly categorized into autosomal dominant and autosomal recessive form. In the past 1 to 2 decades, genomic studies have identified a growing list of genes associated with PKD. Despite the genetic complexity, it seems majority of the PKD genes converge onto the core polycystin 1 and polycystin 2 PKD machinery to initiate a downstream pathological signaling cascade to trigger a range of cellular and organism phenotypes shared across different PKD patients. Patients carrying different genetic variants may manifest variable disease phenotypes onset stage. And although it is important to correlate genotype to phenotype to realize optimal therapeutics, it is financially and clinically more realistic to identify essential pathological pathways that are presented by a wide range of PKD patients. So leveraging patient iPSC reprogramming and gene correction by CRISPR-Cas9, we generated a list of human pluripotent stem cell lines carrying different genetic lesions associated with ADPKD or ARPKD, alongside the respective isogenic controls wherever possible, because there are still 7 to 8% of ADPKD patients genetically unresolved, and we encountered this kind of situations. All these PKD human pluripotent stem cells, for easy, can be successfully differentiated into vascularized kidney organoids, which appears to be quite normal, containing nephron-like structures and vascular network. Spontaneous cyst formation was rarely observed in our system. So empirically speaking, most patient iPSC derivatives do not really present disease phenotype in the absence of stress paradigm. This could be partially attributed to the immature state of these cells or organoids, and upregulation of intracellular CMP levels and the downregulation of intracellular calcium signal represent two critical intracellular hallmarks of PKD, pathological signaling network. So we speculate either upregulating intracellular CMP or downregulating intracellular calcium may be able to trigger cystogenesis specifically in PKD patient iPSC-derived kidney organoids, but not isogenic controls. And this is what we do, either using phoskalin to upregulating intracellular CMP or nifedipine to downregulating intracellular calcium. We observed a dose-dependent cyst formation in ARPKD, patient iPSC-derived kidney organoids, as well as in ADPKD, but I didn't put here. And on the contrary, the gene-corrected kidney organoids, the isogenic control, did not manifest cyst formation in a dose-dependent manner. More importantly, we find intracellular CMP levels positively correlated with the cyst severity, corroborating a critical role of CMP signaling in PKD pathogenesis. When we perform immunostaining of these organoids with nephron segment markers, we perceived in both ARPKD and ADPKD patient iPSC-derived kidney organoids, cyst formation was observed in both proximal tubule and distal segments. So based on a lot of model studies and in vitro cyst studies, we have already understood cyst formation is conferred by a combined effect of apical fluid secretion and epithelial cell proliferation. So indeed, in PKD, iPSC-derived kidney organoids, primary cilia protruded towards dilated tubular lumen, while cyst-lining epithelial cells can potently retain EDU labeling, indicating a very strong proliferative state. The cyst-lining epithelial cells also adopt a sort of dedifferentiated phenotype as represented by the Vimentin staining. However, none of these cystic-related phenotype was observed in the isogenic gene-corrected kidney organoids, suggesting the phenotype must be correlated to the genetic lesion. And cyst formation also leads to a range of pathological phenotypes in kidney organoids reminiscent of PKD. So for example, in the dilated proximal tubules, we could clearly observe upregulation of kidney injury molecule 1, but this signal was completely absent in the isogenic control organoids proximal tubules. And due to the proximal tubule injury, these proximal tubules lost ability to reabsorb dextron in a size-selective manner in the iPSC-derived kidney organoids, but not in the gene-corrected kidney organoids. Most PKD patients develop hypertension, especially towards the later stage of disease pathogenesis. So clinical studies showed in both ADPKD and ARPKD patients, the cyst-lining epithelial cells acquired ectopic renin expression, contributing to the stimulation of renin-angiotensin-aldosterone system. Actually, in PKD kidney organoids, we also observed the cyst-lining epithelial cells acquired aberrant renin expression, whereas in the gene-corrected organoids, we only observed very little renin-positive staining cells localized in the interstitium near the glomerulus, reminiscent of the juxtaglomerular cells. And in agreement, these PKD kidney, this is from AD, this is from AR, and in agreement, ADPKD kidney organoids released more renin into the cell culture medium compared to the isogenic control organoids. To better understand how closely the organoid model represents actual PKD patient's kidney, we performed a single-cell RNA sequencing of PKD kidney organoids and compared the data set with a single-cell data sets generated from ADPKD patient's kidney biopsy by Ben Humphrey's group. So data integration revealed the highest correlation in the proximal tubule and the thick ascending line of loop of Henle between the PKD kidney organoid and the PKD patient kidney samples, supporting the feasibility of using organoid to represent some PKD tissue biology. So this prompted us to compare the single-cell RNA sequencing data set of isogenic kidney organoids derived from patient iPSCs or gene-corrected iPSCs. We hope this could help us to identify some normal disease phenotypes and potentially targetable pathways. So using established cell surface markers identified by many previous studies, our data set revealed 5 major cell types in the kidney organoids, including proximal tubule, distal epithelium, glomerulus, endothelial cells, and stroma cells. Each cell type exhibited the concordant transcriptomic signature and the concordant chromatin accessibility. We also performed the ATAC alongside the transcriptome. So because cyst formation mainly occur in the tubular epithelial cells, we conducted a subclustering analysis of all the tubular epithelial cells, which not only identified proximal tubule, distal epithelial, as well as highly proliferating epithelial cell small cluster. Pathway enrichment analysis revealed the dysregulation of a range of metabolic pathways dysregulated in the PKD kidney organoids compared to the isogenic control organoids, among which we noticed a significant downregulation of autophagy-related pathway. So when we go back to analyze the kidney organoids, we concordantly detected a lower level of phospho-AMPK in the PKD kidney organoids compared to the gene-corrected ones. Meanwhile, LC32 was significantly downregulated alongside reduced number of autophagosomes in the cyst-lining epithelial cells compared to the gene-corrected epithelial cells. This is the statistic analysis. This might be just a association phenotype. So that's the reason why we try to see whether autophagy acting as a critical gatekeeper against the cyst formation. So we overexpressed ATG5, which is a key regulator of autophagosome assembly in PKD patient iPSCs and differentiate them into kidney organoids. And we observe cystogenesis quite effectively attenuated in PKD kidney organoids when ATG5 is overexpressed. Using the in vitro kidney organoid model, we performed a small-scale drug testing and shortlisted a few drug compounds that can effectively inhibit cyst formation in vitro. We already know 2-DG, metformin, and rapamycin can effectively inhibit cyst formation in a range of PKD models, so validating the competence of the organoid model for testing drug efficacy. Additionally, we identified 2 more compounds. This one is adenylyl cyclase inhibitor and minoxidil. Both can effectively inhibit cyst formation in vitro with relatively tolerable dose. And also, we performed a Western blot. It looks like 2-DG, rapamycin, and minoxidil can induce autophagy in PKD kidney organoids, suggesting probably restoration of autophagy may act as a potential means for targeting cyst formation. Back almost 2 years back, FDA announced all candidate drugs no longer all the candidate drugs require preclinical animal study before going to the actual clinical trials. But there are many issues with the in vitro kidney organoid model. It's deprived of the native tissue microenvironment, such as blood flow, systemic hormone. Many scientists also do not approve using the stress paradigm to stimulate the disease phenotype, suspecting about some artifacts. So we wonder whether we could put this diseased human tissue into an in vivo rodent animal to allow some reconstruction of the tissue microenvironment. And this is what we do. We differentiate ARPKD and gene-corrected iPSCs into kidney organoids and engraft the organoid into immune-compromised NSG mice. 4 weeks later, we recover the organoid graft. We find the graft successfully get anastomosed. The glomerula got perfused by the red blood cells. But most importantly, 4 weeks later, we observed a very potent spontaneous cyst formation only in the ARPKD kidney organoid grafts, but in the gene-corrected kidney organoid grafts, it's completely cyst-free. And we also observed the spontaneous cyst formation in the ADPKD kidney organoid grafts. So we are still exploring what happened to these organoids in vivo to trigger spontaneous cyst formation. But in a very simple experiment, we recover the organoid graft to perform explant culture in vitro for 15 days. And this cystic structure can last for a very, very long time alongside the KIM1, the injury marker. But when we compare the organoids to those which were completely grown in vitro, cyst formation triggered by phosclin. And after we withdraw the phosclin, 1 week later, all these cystic structures were completely recovered. So we are still trying to understand what makes this difference, but this might provide us with some suggestion. Maybe the organoid xenograft model is a bit
5
more
minutes
sophisticated
remaining.
in vivo system to represent human PKD. And we also tested we also employed this system to test whether inducing autophagy could help us to inhibit the cyst formation in vivo. And indeed, the doxycycline induction of autophagy, ATG5 in vivo in organoid graft can effectively inhibit in vivo cyst formation, as well as minoxidil in both ARPKD and ADPKD kidney organoid xenograft. But still, we have many limitations of organoid model. One of these we want to target is the absence of immune cells. I will try to be a little bit brief. Because steroid inflammation is occurring in chronic kidney disease arising from various etiologies. And kidney macrophage play a critically important role in regulating the inflammation in CKD. But unfortunately, the macrophage population in the kidney is quite complex, and at different developmental stage and pathological stage, different sources can contribute to the macrophage. But York-Sac-derived macrophage contributed very little to the macrophage pool. That's the reason why we decided to differentiate the human pluripotent stem cells first into monocytes, then followed by into a sort of like embryonic-like macrophages. This macrophage can polarize in vitro to adopt a sort of either M1 or M2 state, suggesting they are functional. And then why we need to introduce macrophage, whether they are correlated with ADPKD. This is a single-cell data set we borrowed from Ben Humphrey's group. Actually, PKD has increased abundance of leukocytes, and also the kidney expressed a significant upregulated inflammatory cytokine or chemokine. So we differentiated PKD patient iPSCs into either kidney organoids or monocyte and co-cultured them as a sort of infiltrating monocyte kidney organoid model. And we observed in the presence of these infiltrating macrophages, cyst formation was substantially upregulated compared to those without macrophage, but we didn't observe this in the gene-corrected kidney organoids. As cyst formation progresses, these autologous iPSC-derived monocytes migrated from the peripheral interstitium area towards the cyst-lining epithelial cells, adhere to the basolateral side of the epithelial cells. While the PKD kidney organoids induce very strong infiltration of these monocytes, the gene-corrected organoids does not possess this ability to trigger monocyte infiltration, possibly due to the lower expression level of MCP1. But you may wonder whether infiltrating monocytes acts only acts as a facilitator, enhancing cystogenesis after cyst formation has already been initiated. But we actually observed something different. When we co-culture the monocytes with PKD kidney organoids, we can actually observe spontaneous cyst formation when we compare these organoids with those without macrophage co-culture. So currently, this project is still undergoing. We are trying to understand whether this pathway could be potentially targetable. So in the end, I would like to say the model has many, many limitations. Although all models are wrong, some may be useful for us to understand some specific aspects of PKD. And with this, I would like to thank my group and my collaborators contributed to all the work I presented here and also our financial support. So thank you for your attention. I'm happy to take any questions.
Thank you very much, Dr. Shia, for that wonderful talk. If you would please put your questions into the chat, we'll go ahead and ask those. And I see that we already have one from Fang. Did you see increased renin release in PKD1 or PKD2 kidney organoids or xenografts also?
Unfortunately, this experiment is really what we want to do, but limited by the resource. We do have an ADPKD family with 4 generations of polycystic kidney disease. Unfortunately, even we performed the nanopore sequencing, we didn't manage to identify any well-known ADPKD genetic lesion. So to answer this question, I do not have a clear answer at the moment, but I hope we could have this done in the PKD1 or PKD2 mutant organoids as well.
Thank you. We have another question from Dr. Muto. Great talk. Are there any candidate cytokines or growth factors secreted by macrophages that promoted cysts?
Well, now we are not very sure, but one thing we could see is in the presence of these infiltrating macrophages, because we are still analyzing the single cell data set, we observe very high IL-6 in the PKD macrophage co-culture, but not isogenic control co-culture.
And Dr. Zimmerman, have you compared your in vitro macrophages to those found at different developmental points in vitro or in vivo?
I didn't have time. Thank you for the question, Heike. Thanks for helping us reviewing many manuscripts. We did. And actually, the monocyte itself is really similar to the PBMC-derived monocyte. And when we introduce into kidney organoid, in the absence of any genetic lesion, so Y type, we find out actually the macrophage identity is very similar to the embryonic kidney macrophage. But this is under the condition of no disease, but not with PKD. Because in PKD, we apply the monocyte topically as infiltrating monocyte, but not like resident. Yeah.
Okay. Next question. Do you see macrophages close to cystic cells or integrated into the cyst epithelia?
Because we apply topically, so they are clawing onto the, I don't know, maybe I'm not sure if my image can, I'm not sure if my image here can show some data. You see the black dots here. Actually, they are topically applied. Basically, this is what you see up front is the basal side of the epithelial cells. We more consider they are clawing on the basal side. Instead, we haven't seen trans-epithelial at this moment. We only see aggressive migration towards especially the very huge cystic structure. So I cannot tell you whether, because you have to really look into every epithelial cystic structure and every single macrophage. But at least for now, what I can tell you is mostly they are clawing on the basolateral side.
Okay. Do you see any change in the polarity of the cyst lining cells in your organoids? Something similar to Dr. Friedman's work.
We still see the primary cilia protruding towards the tubular lumen, but not the other side. Yeah, probably the culture format is a bit different because they are using floating culture, but what we are using is classic transwell culture. So it's static culture. Different culture format may change the phenotype.
Okay. which type of macrophages, M1 or M2, are responsible for cyst formation or expansion?
We are analyzing the data. I'm not completely sure because, we may be questioned whether you see this in the patient, but it is very difficult to obtain this kind of patient samples for analyze. What I can tell is it looks like a TREM2 type B. So it's not a clear M1. M1, M2 is a conventional identification, but there are cells co-expressing both M1 and M2. And when it comes to cyst growth, you have both proliferative. When you have the macrophage to proliferate epithelial proliferation, it might be good for restoring kidney injury, tubular injury, but it's bad for cyst growth. So it's a sort of very rare TREM2 type B macrophage, but it's not M1 or M2.
Thank you, Dr. Shio. We're about out of time here, so if you wouldn't mind answering the remaining questions in the chat, that would be greatly appreciated.
Yeah, I will try to answer their question in the chat. Thank you very much.
Thank you very much. Reminds me of the Oscars. Okay, so I'm pleased to introduce our second speaker of this session, Dr. Nuria Pastor-Solara. Dr. Pastor-Solara earned her medical degree from Jefferson Medical College. She then went on to complete her PhD in biochemistry and molecular biology at Jefferson. Dr. Pastor-Solara completed her nephrology fellowship at the Brigham and Women's Massachusetts General Hospital program. She is currently Associate Professor of Medicine in the Division of Nephrology and Hypertension, and she is also Assistant Dean for Research Mentoring at the Keck School of Medicine at USC. Her talk title for today is Addressing the Therapeutic Potential of Bempedoic Acid in PKD, from preclinical work to the BEAT PKD clinical trial. Dr. Solara, I think you're...
Good morning. I'm just having a little bit of trouble with my computer, so I had to start in the middle. Thank you for that great introduction, and thank you to the organizers for giving us this opportunity to present our work. We had some grant support from Esperion Therapeutics. So we are going to start a little bit with the debate that is it a cyst or an adenoma? We can look at cells of differentiated tissue as undergoing their metabolism to obtain energy through either oxidative phosphorylation or anaerobic glycolysis. However, it has been known since the Warburg effect was described 100 years ago, more than 100 years ago, that proliferative tissues or tumors are able to use aerobic glycolysis. Are you able to see my full screen?
No, you're not in presentation mode.
Still not corrected?
Unfortunately, no. There, we've got it.
Thank you. This work, okay. Thank you. So this work is also that I'm going to present also wants to point out the fact that it's been shown that there was defective metabolism in polycystic kidney disease, work done by Dr. Boletta and others, which was seminal to understanding how we could tackle metabolism to address what was happening in polycystic kidney disease and find a metabolic solution for the disease, so to speak. So in this work, they showed a clear upregulation in PKD1, in a PKD1 knockout system of glycolytic enzymes, such as LDHA and PKM2, as well as HK1. There is also an additionally to having dysregulated glucose metabolism, there's also dysregulated fatty acid metabolism and mitochondrial dysfunction in ADPKD. And this seminal work was done in a number of very prominent laboratories, including finding fatty acid oxidation impaired in a model of ADPKD, and that polycystins were modulated by cellular oxygen sensing pathways, and that in turn they could regulate mitochondrial function. And there was also that a cleavage product of polycystin 1 was a mitochondrial matrix protein that could affect mitochondrial morphology and function when heterology expressed. So our laboratory has been interested in our laboratories group has been interested in addressing how we could modify metabolism to address a number of physiological derangements. And one of the physiological metabolic derangements happens in polycystic kidney disease. So we started thinking, like all of us do in this great conference, thinking about how our patients who have polycystic kidney disease have limited treatment options. And we had been studying a number of pathways, including the AMPK pathway dysregulation in epithelia and other epithelial diseases, but in PKD. So we had also been studying a particular enzyme, ATP citrate lyase, or ACLY, in kidney cancer. And we thought it might be important to check out how this enzyme affected polycystic kidney disease. So we encountered bempedoic acid, which was at the time it wasn't even approved at the time that we identified this compound as a potential therapy for PKD. Now it is approved, and it's approved for the treatment of hypercholesterolemia in patients with atherosclerotic cardiovascular disease who needed additional cholesterol lowering medications. And it was used together with diet and other medicines such as statins for this condition. And it targets HMG-CoA reductase via inhibition of ATP citrate lyase and simultaneous activation of AMPK. Interestingly, this enzyme, pharmacological agent is only able to get in the cells through a particular transport protein, which is unique to the kidney, is not present in muscle. So therefore, as opposed to these other medications called statins that have muscle toxicity, and that are also being tested in PKD with clinical studies, we thought it might have a better safety profile. So we hypothesized that bempedoic acid could be beneficial in ADPKD by inhibiting cyst growth, inflammation, injury, fibrosis, and metabolic dysregulation via ACLY inhibition and AMPK activation. We also hypothesized that bempedoic acid could have additive beneficial effects when combined with what now has become the standard of care, and especially for patients who have rapid progression, which is the tolvaptan treatment. Therefore, we proceeded to study whether ACLY activity, which is the target of bempedoic acid, was increased in polycystin-1 kidney epithelial cells versus control. And this was work done by Dr. Hoi Lee, longstanding member of our lab. And we saw an increase in ACLY expression in both proximal tubule-derived and collecting duct-derived PKD cell models. We then proceeded to study the effect of ACLY inhibitors, including bempedoic acid, abbreviated here as BA, on 3D cultures of proximal tubule and collecting duct-derived polycystin-1 knockout cells. And this was work done by Dr. Biagio Saita and as well by Dr. Hoi Lee. And we can see that there is a significant decrease in the size of the cyst in both cell models. We also, and this has been published in this paper that I've been mentioning for the last few slides. And then we also showed that ACLY knockdown significantly inhibited cyst growth in this same type of cell model. And we used the PKD1 heterozygous cell model and as PH2, as well as the PKD PN24 cell lines. And these cell lines were graciously provided by the PKD community. We didn't generate them, so I want to acknowledge our, the fact that the PKD community is sharing their reagents so graciously for people who need them for their studies. Continuing with the same study, we now used a mouse model obtained from the PKD RRC, which is a rapid induction model with doxycycline. And we treated these mice with the help of Jonathan Wong and Samar Sefer, Valeria Mancino, and Jessica Pham, who are mentioned here and shown here. And we showed that there was the treatment with bempedoic acid was able to, in combination, alone or in combination with tolvaptan, was able to significantly reduce the size of the cysts that were obtained by 22 days when the animals reached the control animals reached sort of end stage kidney disease, and the treated animals had better parameters indicating less severe kidney disease. So we also found that bempedoic acid treatment reduced the expression of the proximal tubule injury marker KIM1. To a lesser extent, it also worked on the kidney injury marker NGAL, which is more of a distal injury marker. And we saw significant reduction with bempedoic acid, as well as bempedoic acid and tolvaptan having an additive effect on reduction of the injury. And then the effects of bempedoic acid and tolvaptan in these mice also had an effect on liver signaling. And as expected, we saw that bempedoic acid reduced ACLY activity and stimulated AMPK activity, as well as inhibited mTOR and ERK signaling pathways. And this work was done in immunoblots. Our findings suggested that bempedoic acid may prevent hepatocyte injury in the setting of high-dose tolvaptan treatment, which is always a worry in the clinic. Moving along, now I show unpublished results of bempedoic acid treatment in also a model obtained from Maryland. And we're very thankful for that. But this model is a model that is slow inducible PKD1 knockout model. And you can see here that at postnatal day 60, controls have cyst, and at postnatal day 120, control animals also have extensive cystic disease, and how bempedoic acid reduced that in the histology in panel B. And then we did total kidney weight to body weight, and we also saw that 120 days there was a significant reduction in this parameter in animals treated with bempedoic acid. We then proceeded to look at fibrosis using serious red staining and image J, because once there is fibrosis in the kidney, it is believed that pretty much the damage is reversible. And what we saw is that bempedoic acid versus control treatment, a significant decrease, with statistically significant decrease of the fibrosis index with bempedoic acid, which is in around, and we tried to measure pericystic fibrosis. So the control mice still had cysts, but they had less fibrosis around the cysts, which I found very encouraging. Then also bempedoic acid also slowed cyst growth now in a PKD2 null cell culture. So with a lot of encouragement from Dr. Jin Shi, we started here at USC, we started organizing and participating in clinical studies, which traditionally, even though we are a group of clinicians, physician scientists, we hadn't done. So we have here the conclusions that bempedoic acid inhibited cyst growth in vitro and decreased ADPKD severity in vivo, improved cell injury, proliferation, mitochondrial dysfunction, and that these effects were likely mediated via ACLY inhibition and AMPK activation. So we thought that combining bempedoic acid with tolvaptan treatment further improved ADPKD outcomes in vivo, reduced cyst growth, injury, and other disease parameters in the mice, in the mouse models actually. There was also beneficial effects of bempedoic acid in pericystic fibrosis, very encouraging. And we thought that bempedoic acid may represent a promising new ADPKD therapy having beneficial effects, either alone or in tolvaptan, and this was shown by work in mice. The advantage was that the FDA had approved bempedoic acid for the treatment of this hypercholesterolemia, and so we started preparing for a potential clinical trial. We were very thankful to the group that we first reached, and then all the groups that are now participated in the creation of this clinical trial, which we call the bempedoic acid therapy in polycystic kidney disease randomized clinical trial, abbreviated as the BEAT-PKD trial. So initially we approached Dr. Watnick and Dr. Seliger, and then Dr. Perrone, and then the Pittsburgh group with Dr. Abebe, and then Drs. Craig Gordon and Dana Miskellin at Tufts, and then Dr. Steve Seliger, of course, in Maryland. And then we also had here at USC, Dr. Vinay Dudalwar, who's an expert radiologist and expert in abdominal radiology. So we proceeded with formulating the hypothesis that the bempedoic acid is safe and tolerable in individuals with ADPKD, and repurposing bempedoic acid used for this group would be beneficial in improving ADPKD, whether the patient was or not being treated with tolvaptan. And our first aim was to run and establish this placebo-controlled trial to determine whether bempedoic acid at the dose of 80 milligrams, which is the usual dose, once daily versus placebo, was safe and tolerable over 24 months in a sample size of 120 individuals with ADPKD. That these individuals would be that we would select for the study would be at high risk for progression with or without concurrent tolvaptan therapy. We also said that we would assess the effects on changes in estimated GFR, and then the biomarker that we have right now, which is total kidney volume, height adjusted, and as well as we would look at liver volume, and more sort of experimental or exploratory outcomes such as kidney and liver cyst volumes, and visceral abdominal fat, about which we heard a little bit yesterday from the group in Colorado. And the second aim, we would proceed to measure selected metabolic, inflammatory, and injury biomarkers on the urine samples from participants before and after the initiation of the study drug to evaluate potential correlations of ADPKD severity and response to bempedoic acid. We created this brief description of the steps in the trial with the screening time and then the treatment time. We also we're very thankful to Rina Tarchian here at USC and the pharmacy committee at Maryland for figuring out how to formulate the placebo, which had been an issue for us for a long time. We were unable to obtain placebo. So what I want to point out is that there is a number of in-person visits, and then there's some lab visits, but that we are really, what's really important to us is that the standard of care is maintained in this trial. This is busy, and I don't mean to point out all the different steps. On the left, you see the table 2 for our proposal that was sent to the Department of Defense. And this proposal has been recommended for funding at this stage. And so it's a busy study where the patients get also imaging, as we said earlier. But I also wanted to point out that we're getting the urinary biomarkers as well as MCP1. And these biomarkers are similar to the biomarkers that were presented by the TAME PKD study, which looked at metformin in this disease. And here I have the sort of organizational chart for the study, which is the principal investigator is Dr. Ken Hallows. Our research group is moving to University of Vermont Medical Center, and the radiology group remains here at USC, where Dr. Dudalwar will be doing the analysis of the organ volumes and the and as well as the visceral fat. And then we have the clinical sites in Burlington, Vermont, at the University of Maryland, and at Tufts, with very important, the data coordinating center being at the University of Pittsburgh with Dr. Abibi. So I just want to sort of advertise, we need 60 participants per study arm. We're gonna be, actively recruiting for this study, and we will announce when the study actually begins. It's planned that the study will begin getting established sometime in this summer, and we have 60 participants per study arm. The adverse events we'll be looking at, this is important, would be hyperuricemia, because bempedoic acid has been associated with it, gout, liver transaminitis, and worsening anemia. And then we're also going to be looking at health-related quality of life metrics with a number of questionnaires looking at pain, discomfort, fatigue, emotional distress, and impaired mobility. And I just want to acknowledge our collaborators and really this community. We are very thankful to our patients and the patient community here at USC, the PKD Foundation, and Dwight Odland, who's a patient advocate in our clinic and has really encouraged us to move from the bench to the bedside. I already mentioned USC, Dr. Dudalwar, Maryland, Dr. Wadnick, Dr. Seliger, as well as the pharmacy, and Tufts, Dr. Perrone, and the site PI, Dr. Miskilin, and as well as Dr. Craig Gordon and Dr. Abebi. In our labs, I already mentioned the Ken Hallows lab, Hoi Lee and Valeria Mancino, Jessica, and then a medical student who's now already a resident, Polly Huang, who helped with some of the mitochondrial work. And in my lab, Dr. Saita, Dr. Samarchak Reza, and Dr. Saman Safer. We also want to acknowledge Dr. Anji Chong's lab, where helped us with the administration of bempedoic acid and in the rapid model, and as well as Dr. Zhongwei Li. I didn't present the data, but we're collaborating with him. And we have to thank Aspirion for the providing us with pharmaceutical grade bempedoic acid. And I'll take any questions.
Thank you very much for your wonderful talk, and congratulations on being recommended for funding. We have a couple of questions in the chat. First one, ADPKD patients often develop hyperuricemia, which is the most common side effect for bempedoic acid. What are your thoughts about that?
Yes, so we have to be very careful about that, about including patients who have had sequelae. You know, we were able to obtain you know, an FDA IND exemption for the use of this compound, but we will have to carefully evaluate patients who have had hyperuricemia or gout for inclusion, and then you know, as a potential condition for withdrawing from the study, because patient safety is always first.
Next question is from Dr. Alan Yu. How does the effective dose in mice compare with the proposed human trial dose?
That's a very good question, because it's not like we, basically we're just going to be obtaining the medication from our investigational pharmacies and then preparing the placebo as well. So what we did initially is we, with the collaboration of Dr. Pinkowski, we actually, the way that the mice were dosed was with a comparable dose with what the patients receive. So that's how we did the mouse studies, where by using the drug for the mice that would be comparable to what a patient, a human takes. So so basically it's comparable, and it's based on all the other animal studies that were done for the other indication for this medication.
Okay, thank you. Next question, can you please elaborate on how cholesterol synthesis inhibition with bempedoic acid inhibits fibrosis? And does the drug treatment in your model also reduce inflammation?
So the inflammation part is important. We think it does, but didn't present any data. As far as the inhibition of fibrosis, we think it also has to do with the inhibition of the inflammation. So we do, the question is, are we seeing less inflammation because the cysts are maybe not as large, right? I presented a lot of data saying that the cysts were not as large. And or is the bempedoic acid, by preventing, by changing the metabolism, changing transcriptomal, the transcriptome in the cells, and then there is a less expression of neoantigens that can sort of call the immune infiltration into the kidney. We don't know that. We do know by studying the collagens that are expressed in the cell culture models that the types of collagens expressed by the cystic cells in the polycystin 1 knockout models, that the types of collagens, the abundance of certain collagens that are considered developmental types of collagens change
1 minute
compared to cells that are not cystic. Thank you so much. I heard that. So we do know that there is a change in gene expression depending on collagens, depending on the stage of the cyst, if you want to call it. like that. And we don't know if bempedoic acid is actually having a transcriptional effect or it's having an effect on the general process of the growth of the cyst due to changes in metabolism, and that's why the collagens are different. That was a long answer.
Well, thank you. I think we don't have time for any additional questions, but there are others in the chat, if you wouldn't mind addressing those.
Thank you very much.
Thank you. And with that, I'll turn this over to Mene.
Thank you. Thank you, Dr. Mesa, and good morning, everyone. My name is Duamine Yimano Omene, and it's my pleasure to introduce the next speaker, Dr. Eduardo Chini. Dr. Eduardo Chini is a professor of anesthesiology and pharmacology at the Mayo Clinic, and among many research interests, Dr. Chini has shown that dietary manipulations such as caloric restriction can ameliorate ADPKD progression. He described the role of the ectoenzyme PAP-A in the pathophysiology of ADPKD and identified it as a potential pharmaceutical target. Dr. Chini's talk is titled Discovery of the Effect of Caloric Restriction in ADPKD, Leading to the PAP-A Story.
Can you guys hear me?
Yes, we can.
Okay, can you guys see my slides?
Yes, I can.
Okay, all right. Well, so thank you, number one, for the invitation. It's not very often
you need
that
to
I
swap
talk.
your displays. Excuse me. Swap your displays. We're seeing the presenter view.
Okay. Is it better? Hello?
Yes, it is.
Okay. All right. Okay. So again, not very often that I talk to the PKD folks. We kind of do this on the side on my lab. My lab's more dedicated to metabolism and age-related diseases. But I think that very obviously we have done seminal work that is implicated for polycystic kidney disease, and I change a little bit the title. So lessons learned from nutritional manipulation in polycystic kidney disease, and then that leads to the PAP-A story. So number one, here are some financial disclosures. So we have a patent file for PAP-A inhibition for treatment of APKD. We receive funding from industry, and we've been funded by the PKD Foundation. Unfortunately, we have never received any NIH or DoD grants for this. So what I'm going to talk about today is to discuss a little bit of the basic scientific discoveries behind the opportunities in nutritional manipulations in polycystic kidney disease, and then talk about how we actually come up with this idea many years ago, over 15 years ago, and then talk about how did that discovery led to the PAP-A story. And just to also fulfill you guys a little bit, I'm a physician scientist. I'm a transplant anesthesiologist. I see a lot of patients having solid organ transplants. And part of the idea to go back to PKD was based on some patients I saw in the OR, and also the fact that I work with Tom Doja, that was one of the mentors of Dr. Vicente Torres for his training. So my lab is a basic science lab that has the goal to understand the basic biology and basis of mechanisms of disease. And we also go all the way now to translation, and we have been actually quite successful with this in different things. So why did back over almost 20 years ago, I thought about the idea of trying caloric restriction in polycystic kidney disease. Essentially is because almost every single pathway that is fighting to regulate cystogenesis and the disease process in PKD are mediated by metabolic sensors. So as you guys know, AMP kinase, mTOR pathway, mitochondrial dysfunction, and there's some not extremely strong evidence for sirtuins, but they are all metabolic sensors. And my lab dedicated, as I said, to metabolic diseases and especially aging and age-related metabolic dysfunction. And these are exactly the same pathways that have been implicated in metabolic dysfunction during aging, and many of those have been approached to change the course of aging and longevity. And obviously, as you guys know, caloric restriction is one of the few things that has been clearly demonstrated in animals, and there is evidence of metabolic reprogramming in humans with caloric restriction. So we decided to do this. And coming from outside, I didn't have any preconceptions. And in fact, when we first talk with a couple of people saying, well, there is clearly a metabolic component here in PKD, the majority of people didn't actually believe and think that there was nothing metabolic about PKD. So this is then the paper that we publish in the Journal American Society of Nephrology. It has, over the years, received a lot of attention. It's one of the 5% most cited papers and highly cited on the field. So what we described was that restricting food or caloric restriction ameliorated development of polycystic kidney disease in animal models. And this was done by Gina Warner. That it was the most amazing research technologist I ever had in my lab. She was able to conduct research completely independent and work as a research scientist, essentially. So what we did was taking the RCRC mouse that was developed by Dr. Harris at Mayo and Dr. Torres, and then we put them on caloric restriction. And as you guys can see in red, and this is all published, those animals don't gain as much weight. And then when we did ultrasound, you could barely detect any cysts. Those kidneys were obviously smaller, and then by histology, there was less cysts. By staining for fibrosis, there was certainly less fibrosis, less inflammation, and less evidence of injury. Metabolically, there wasn't a lot actually of changes happening, except for the expression of exokinase 2 that was upregulated in the animals that had PKD and were at libidin diet, and this was resolved by caloric restriction. The really interesting thing was that this was not only a way to prevent polycystic kidney disease, but also it could reverse. So here in red is the curve of the kidney to heart weight changes over time. So up to 7 months, there's this increase in the kidney size. And then after that, there's a decrease, mostly because of fibrosis. So if we started caloric restriction at a very early age, or if we started caloric restriction after the kidneys were already cystic, we could actually ameliorate not only preventively, but also in terms of treatment. And we actually showed that this was dose dependent. So if you go from, so the data that I showed before was all at 40% of food restriction, but we could actually do 10 20%, and showed that this was actually dose dependent, both at the gross anatomy and also at the microscopy. We went on to actually demonstrate that this was at least partially mediated by an activation of the AMP kinase LKB1 pathway and inhibition of the mTOR pathway, potentially leading to the decrease in cystic growth. It's obviously hard for you guys to read, but after we published our paper, there were several other follow-up papers by different groups demonstrating that indeed caloric restriction could ameliorate polycystic kidney disease, and there's evidence that actually the weight loss is important for that to happen. We actually apply for funding for this, and were, as I said, never successful for some reason. We went on to do a second study where we actually went on and decided to do amino acid restriction. So we did methionine restriction that is kind of a vegan kind of diet, and that demonstrated with collaboration with Vishal Patel's group that this also protected kidneys from developing cystic disease. So this is a paper that was spearheaded on our side by Sonu Kashyap, and we were co-senior authors on this. So this is obviously a very, 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 very busy slide, but I'll have some follow-up slides that demonstrate what we did. So essentially, we did this methionine restriction. So it's a single amino acid restriction. There is some decrease in body weight, but certainly not the same as what you see with caloric restriction. The food intake is actually very, very similar between the two groups. And in fact, if you adjust by weight, the animals on methionine restriction are actually eating a little more. We also did some metabolomics on the PKD kidneys, and we found that actually methionine levels were increased. And that's actually one of the reasons also why we went on and decided to do this methionine restriction. When we actually did the methionine restriction, levels of SAM, this methyl donor that is synthesized from methionine, decrease. And Dr. Patel's group demonstrated that methylation of RNA was very important for the development of cystic disease. As you guys can see here, so when you actually do the methionine restriction, this is a control ADPKD kidney. Here are the methionine restricted, so they're smaller kidneys. There are less cysts, and we show also less injury. So, as I said, this was then followed up by many other investigators, and there was a study that correlated then that BMI and cystic growth kind of go hand to hand, and perhaps loss of weight in patients with PKD would be a potential way to decrease the progression of the disease. And obviously, caloric restriction is one of the ways, and there are, now anoretic drugs like GLP-1 receptor agonists. And we are doing some work with GLP-1 receptor agonists, with a caveat that those compounds also increase cyclic AMP. So it's kind of a complex interaction there. So there are now multiple clinical studies looking at the effect of metabolic manipulation. I'm not sure when, perhaps this was shown yesterday, but there was a trial on the caloric restriction for PKD. So our then follow-up study, we we know that adhering to caloric restriction is very, very difficult. So we decide to try to understand mechanistically what was caloric restriction doing, and if we could find that, we could then potentially find a specific target that we could manipulate. So we then published this paper in JCI Insights. This paper was under review in Science Translational Medicine for 2 years when it was finally rejected. And then we got, in just a week, accepted in JCI Insights. So the idea here is to, as I said, to look at what does caloric restriction does, and we knew that the IGF pathway is very, very important for the effects of caloric restriction, and also the IGF-1 pathway regulates the mTOR pathway, and it's implicated in aging and longevity. So we went on and looked at this pathway. So there is some evidence in the literature that IGF pathway could be implicated in PKD, but it wasn't until our paper in JCI Insights that there was clear evidence that actually the IGF pathway was actually instrumental for cystic growth. So what we did, and actually to go to this this slide here first, it's a poor reproduction. This is actually from the supplemental material on this JCI Insight paper. But what we did here is we took in black on the bottom on D the wild type animals, and then we look at microarrays, and then we did PCR to validate. But this is levels of PAP-A are, you know, tenfold higher in the PKD RRC or C mice. And then when we actually put the animals on caloric restriction, in blue, the levels of PAP-A decrease. And levels of other components of the IGF pathway also decrease. It's important that the IGF-5 is actually a marker of activation of the IGF pathway, so that indicates that the activation of the IGF-1 receptor, or you know, depending on the receptor, it's decrease. So here's part of the study that then we did. So as I said, in PKD RRC or C mice, there's more PAP-A, there's more of other components of the IGF pathway, and this actually correlated extremely well with the size of the kidneys. We also have data showing that in pregnant mice, levels of PAP-A and IGF components are increased, and there is in mice. Obviously, I'm not saying that that's true in humans, but in mice, there's an extremely increase in kidney growth and cystic index when you actually do analysis of multiparous mice. The PAP-A keep going up over time, so it's not one of the drivers of, I mean, there's no specific mutations of PAP-A or IGF pathway, but it is necessary for cystic growth. I think there's this obsession about something being mutated on a disease should be a target, and as we know that, you know, the only therapy approved for polycystic kidney disease is not indeed mutated to the AVP receptor. So we went on to actually demonstrate that this was actually mediated by the increase in PAP-A, so it was mediated by a cyclic AMP dependent pathway. I'm not going to show tons and tons of data. This is on the paper, but this is actually animals here. So this is wild type animals. We actually gave one shot of phospholine IP, and then we look at PAP-A in the kidney, and there was no change on PAP-A in the kidneys of the wild type, But if you actually go and give it to the RCRC mice, then there is an increase in PAPA expression. So it appears to be actually specific to the polycystic kidney disease mice. We also look at normal renal cells versus L12 cells. They are derived from PKD patients. And again, if you actually give phoscolene, there is no expression almost of PAPA, but if you do that on the 912s, it does. We also look at this animal model that was developed by Dr. Torres that there is an increased activity of protein kinase A, and obviously those kidneys are extremely cystic, and on that model again the levels of PAPA were increased. So we went on and then crossed the RCRC mice to the PAPA knockout mice. And as you guys can see, there's a dose-dependent effect. So if you actually have the homozygous for the knockout PAPA, so they have no PAPA, there's almost no cysts, and those kidneys are pretty normal. If there is heterozygous, then the kidneys are smaller. And you delay the progression of the disease. So again, as you can see here in the histology, 2.5 months, 4.5 months, and 12 months, the heterozygous delays, eventually you catch up, but with the knockouts, those kidneys almost never get to a full cystic size. And we actually follow up those animals and show that the PAPA both heterozygous and homozygous RCRC lived longer and had a delayed development of renal failure. We also show that in cystogenic assays in vitro that you could actually grow cysts better when you actually combine phoscolene with IGF, and that when you actually give phoscolene to a metanephron in culture, you also increase PAPA expression. So mechanistically, then we look at metabolic sensors, and again, when you actually have inhibition of PAPA, you have decrease in activation of the IGF receptor. You have decrease in cellular proliferation, inhibition of ERK, and then phospho-AKT, and activation of the cyclic of the AMP kinase pathway. So we went on to look at the therapeutic approach. So my lab does a lot of work with ectoenzymes.
5 minutes remaining.
Okay, thank you. With ectoenzymes, so we actually took then antibodies against PAPA and tried to see if that work in models of PKD. And indeed, using a commercially available antibody against PAPA, we actually demonstrate. So here's wild type animals. Here's the RCRC receiving vehicle, and here's the RCRC receiving the anti-PAPA antibody that blocks its activity. It's not the cytotoxic antibody. So you have decrease in kidney to heart size, decrease in cystic area, and then you have a decrease in inflammation and markers of injury. So if you actually look at patients with PKD, we actually saw that there was an increase in actually PAPA in the cystic fluid of patients with PKD. There was an increase in expression of PAPA, looks like in the cystic lining. And then if you actually look at mRNA levels, especially IGF-1 is quite increased in patients with PKD. So in terms of this new technology that everybody has to show now, we look at the expression where which cells are actually expressing PAPA, so control in PKD patients, and it looks like it is on the distal nephron where I guess it does make sense. There's actually an increased opening of the chromatin on these areas of the PKD patients, so it makes sense there's more expression. So in conclusion, ADPKD has an important metabolic component. We were the first ones to actually show that nutritional manipulation like caloric restriction can actually ameliorate PKD, not only prevent, but also treat. And mild caloric restriction, even only 10 to 20% of caloric restriction could potentially be safe and feasible for patients with PKD. Obviously there's a trial that's being concluded. And then we validated those, hopefully there will be validation of those basic discoveries, both with caloric restriction and also with PAPA inhibition in the near future. So just to conclude, Fouad Shabib, that's now here in Mayo Clinic, Florida, has proposed this algorithm that now includes that in obese patients, caloric restriction under medical guidance should be considered for patients with PKD. And I'm very, very happy to be a non-nephrologist that has contributed to a potential therapy for those patients. So any questions, I'll be happy to answer.
Thank you, Dr. Cheney. There's a question in the group here. It says, PAP-A knockout blocks cleavage of all PAP-A substrates, IGF B2, 4, 5. PAP-A antibodies more selective to IGF-BP4 cleavage. What differential impacts do you speculate from these two different approaches? Which IGF-BP substrate is most important in regulating IGF signaling in kidney and ADPKD?
So it's more likely to be 4. So as I said, 5 is actually more a marker of activation. So there are many other layers also to the PAPA pathway because there's also endogenous inhibitors like the stannocalcin. So I think those are things that we actually propose on a grant to be explored. Again, we have been unsuccessful, but there's much to learn there. But yeah, I agree. I think that figuring out not only which specific substrate you need to block its cleavage, but also in which location and which cell, I think it's very important.
Okay, thank you. Please, if you have any questions, post it in the group chat. I do have one. PAP-A levels are used clinically to screen for Down syndrome in pregnant women. Do you think that, or can you speculate as to whether it can be used as a biomarker, whether diagnostic or prognostic biomarker, or even therapeutic biomarker in ADPKD patients?
Yeah, so we are exploring that. So we are looking not only at PAPA, but also multiple components of the IGF pathway. And we do have some evidence that the IGF-BP5 as a marker of IGF pathway activation is potentially an indicator. PAPA has also been used in also other components of the IGF pathway to be explored as markers of renal failure itself. So there is this obviously difficulty there, what is just cystic growth and what is kidney dysfunction. But yes, this is something that we're exploring in the lab.
Okay, good, thank you. We're still waiting for other questions. I'll just ask one more then. You described the role of PAP-A in ADPKD. Is it also like the upregulation rather of PAP-A in ADPKD? Do you also see or speculate that it will be upregulated in other cystic diseases like ALPKD?
So we have not done that. I think it is certainly possible, but we have not done that.
Okay. Yeah, there's another question here which says, would you expect a similar beneficial effect of PAP-A inhibition or knockout in on liver cyst?
Yes, certainly. And in fact, I think that there is some microarray data that I think demonstrate that actually the IGF pathway is one of the top hits for polycystic liver disease.
Okay. Thank you. I think there are no other questions, so I think we can move on to the next speaker. Thank you. Okay, so the next speaker is an abstract presentation, and it's titled Alleviating the Kidney Cyst Growth Through Genetic and Therapeutic Targeting of Methylation-Controlled J Protein. And this is by Marie-Luce T. Monaghan.
Just sharing my screen. Okay, thank you very much. First, I'd like to thank the RRC for selecting my abstract. My name's Dr. Mary-Louise Monaghan. I'm a postdoctoral fellow at the University of Colorado in the lab of Katharina Hopp, and I'm going to talk to you today about alleviating kidney cyst growth through genetic and therapeutic targeting of methylation-controlled J protein. So multiple studies have highlighted that dysregulated metabolism is a key feature and disease modulator of ADPKD. And this story really started with the Wahlberg effect and the work from the Valletta lab, where it was found that the PKD epithelium predominantly utilizes aerobic glycolysis, and that glycolysis inhibition with 2-deoxyglucose slowed kidney cyst growth in rodents. Now, if we consider mitochondrial metabolism more specifically, ADPKD models have been shown to present with reduced fatty acid oxidation, and work from the Patel lab found that treatment with PPAR alpha agonists actually enhanced fatty acid oxidation and reduced cyst growth. And lastly, transcriptional analysis of ADPKD patient cells and mouse kidneys are indicative that oxidative phosphorylation is also impaired. So considering these entities, we mined some single-cell RNA-seq data that we derived from the RCRC model, which we know is a hypomorphic model orthologous to human disease. So here we have UMAP data of the collecting duct compartment, so the non-proximal tubule clusters. And we identified here cluster 4, this unique cluster that was highly expressed in genes known to be altered in PKD. And we can see here with the PKD signature. So taking cluster 4, along with clusters 0 and clusters 2, as they were the most related gene signatures, pathway analysis revealed multiple dysregulated pathways. And of note here, we have oxidative phosphorylation, electron transport chain, complex 1 biogenesis. All of these were found to be downregulated in the unique PKD gene cluster that we identified in the RCRC model. We also wanted to determine if there was a change in energy charge in our CRC mice. So through purine metabolomics on SNAP frozen kidneys, we found that the ADP-ATP ratio was actually significantly reduced in our CRC mice compared to wild type. And I do want to highlight this as pilot data, so we do have relatively low ends throughout, and it is an ongoing study. So considering this, if we circle back to the concept of mitochondrial dysregulation or mitochondrial metabolism being dysregulated, we also know that there's mitochondrial structural abnormalities prominent in the ADPKD epithelium, such things as swollen and abnormal cristae, decreased mass, and reduced fusion. We also know that PC1 and PC2 have been shown to localize to the mitochondrial-associated ER membranes. And work from Kaplan and Germino have suggested that this C-terminal translocation of PC1 to the mitochondrial matrix modulates things like tubular and cyst cell proliferation, metabolomic profile, mitochondrial function, redox state, and has been shown to actually suppress cystic phenotype. So therefore, considering the role of the metabolic mitochondrial metabolism, we wanted to think about how the electron transport chain might be impacted and affect this. And this took us to work out of Iowa, where this group here employed 2 models of ADPKD, a rapid and more slowly progressive model. And using Seahorse, they measured oxygen consumption rate across the 4 complexes of the ETC and found that compared to wild type, they were downregulated in both models. And this also mirrored complex activity across all 4 complexes in both models. So considering the role of the ETC and the fact that the complex activity was down, we turned to a collaborator of ours, Dr. Mercedes Rincon, who's studied the electron transport chain extensively and has identified methylation J controlled protein as a negative regulator of the electron transport chain. So MCJ is a small nuclear protein and it translocates to the inner mitochondrial membrane, binds to complex 1 and reduces the activity, acts as a natural inhibitor of complex 1 and the electron transport chain. Loss of MCJ increases complex 1 activity and the activity of the electron transport chain, as well as mitochondrial membrane potential, increases oxidative phosphorylation and ATP generation. This, as a result, improves mitochondrial respiration and metabolism, but it doesn't increase that of ROS because the loss of MCJ actually forms supercomplexes that prevent minimizing electron leakage and stop that buildup of ROS. It's important to note also that MCJ does not impact physiology or the lifespan of mice, and the absence of MCJ in vivo has been shown to alleviate various types of liver disease. There is also an siRNA to MCJ, and it mimics the pathophysiological benefits of the genetic loss of MCJ. It's actually currently been evaluated for both toxicity and efficacy in non-human primates for IND enabling, particularly for the treatment of NASH. And if I draw you down here, we have looked at this in wild-type kidneys and found that the naked delivery of the siMCJ is enough to knock down MCJ protein levels in wild-type kidney. So considering all of this, we wanted to find out how well expressed MCJ is in the kidney and in ADPKD. So we mined published and in-house single-cell RNA-seq data. And starting here, we have a data set from the Humphreys lab published in Nature Communications and looks at human ADPKD. And we found that it was well expressed across many kidney compartments. And of interest, it was actually upregulated in PKD in compartments such as proximal tubule frail repair and proximal tubule. We then mined our own single cell data of our CRC mice and found again that MCJ was well expressed across the kidney, but there wasn't the change in expression that we've seen in the human data. This was then backed up from the Zimmerman lab with spatial transcriptomics, where again you see MCJ is well expressed, but there doesn't seem to be a clear difference between wild type and our CRC. So we wanted to then ascertain a role for MCJ in ADPKD. And as such, we decided to cross the RCRC mouse with a global MCJ knockout. And here you can see from the histology, there is a significant reduction in cyst burden in the knockout of MCJ. This was significantly mirrored in kidney weight, body weight, and computed cyst volume, but it did not hold significantly in computed fibrotic volume, but there was a downward trend, showing that there is a lessening of disease burden in the knockout of MCJ. I also want to highlight here the delta % difference. In females across all 3 matrices, there seems to be a more significant reduction in disease burden, suggesting that there is a sex difference in this model. So given that we'd seen this alleviating of disease burden, we also wanted to confirm if there was a change in metabolic state. So herein we used FLIM, which is fluorescence lifetime imaging microscopy, which allows us to manipulate the half-life of free and bound NADH to determine whether glycolysis or oxidative phosphorylation is more prominent. So more free NADH indicates glycolysis, whereas more bound NADH indicates oxidative phosphorylation. So in the wild type for MCJ model in our CRC, the yellow staining indicates more glycolysis. When we knock out MCJ, we see a shift, more magenta shown, and that's indicative of oxidative phosphorylation. So given that we did see this shift in metabolic pathways, we wanted to confirm if ROS levels held steady as predicted.
2 minutes remaining.
Herein we used electron pragmatic resonance, and we found that the levels of ROS remained stable in the RCRC model between wild type and knockout MCJ. If anything, there's a downward trend in the knockout, but we did confirm that ROS levels were in fact upregulated from normal kidneys, wild type kidneys to RCRC kidneys. So one more part of data I wanted to discuss is I've also performed global kidney metabolomics, and it seems that MCJ loss does correct metabolic dysregulation characteristic to the PKD kidney. So if we look at the dysregulation between wild type kidneys and then to the RCRC MCJ wild type, the dysregulation viewed in that change seems to be corrected when you knock out MCJ. And that's specific to amino acids, fatty acids, citrate, and succinate and ferminate are all entities that I found to be corrected in the MCJ loss. This is very new data and it's something I'm still mining, but I do think it's quite exciting given the rest of our data. So to conclude, MCJ is an attractive target for the treatment of ADPKD given our preliminary findings. What I'm thinking of moving forward with this project is that I think it's important functionally to determine what the electron transport chain complex activity is following MCJ loss. And I think it would be interesting to look at that across different cell types. Is there cellular differences between cystic and non-cystic or epithelial and non-epithelial? Is this MCJ loss universally affecting all electron transport chains within the kidney? We are currently in the process of starting a preclinical trial of SI-MCJ, and we'll be doing that in both a rapid and slowly progressive model of ADPKD to confirm that the therapeutic knockout mirrors that of the genetic knockout. And as I've shown in the data, we have not cured ADPKD here, but given the multifactorial nature of dysregulated metabolism in ADPKD, I think considering MCJ along with combinations such as 2-deoxyglucose should be interesting, and just want to thank all the co-authors, particularly my PI, Dr. Hop and Dr. Mercedes Rinken. This work was funded from an RCRC pilot and feasibility grant awarded to my PI, which we're grateful for, and along with my PKD Foundation Fellowship and my boss's active DoD grant. And I'll be happy to take any questions.
Excellent talk. Thank you very much. Is there anyone have a question who can post in the group? So in this ADP in this model that you used, did you look at the this is the RCRC model? Did you look at any improvement in the liver or and look at the mitochondria morphology in this model?
We have the liver histology and the liver frozen tissues, and it is on our to-do list to look at any dysregulation in the liver based on some early data we've seen elsewhere.
Okay. Okay, there's a question here from Dr. Watnick, and it's asking if we are planning to look at other mouse PKD1 model, PKD1 or 2 mouse models.
At the moment we are focusing on bringing it into a more rapid developing model, so looking at the RCRC and the biopsy, but we don't have any plans at current to look at it in different models, but I could see it being something we looked at further down the line when we're out of the more preliminary data gathering stages.
Okay. And there's another question is asking whether you speculate any potential side effects of silencing MCJ.
I think this is something that if any point you're silencing a protein, you need to consider. I think I know the work that's went on in liver is extensive and has been going on for years. So I do feel like we have good grounding in the literature to suggest it is a safe thing to target, but it obviously is something I'd need to be considerate of at all points during it.
Okay. In your proposed mouse study using the MCJ siRNA, when do you plan to start the treatment during the disease progression?
So the trial we're getting underway just now with the biopsy model, we are going to intervene pretty early in disease, probably 8 weeks old, with the given how rapid the disease progression is. So I think it is currently we'll take the data from the biopsy and make a decision going forward for the more slowly progressive model, but I would imagine it would be a relatively early intervention.
Okay. There's another question from Dr. Alan Yu, which is asking, are there any adverse consequences from upregulating the electron transport chain?
So again, similar to the is it safe to silence MCJ, why, sorry, I was part reading a question. I think you are manipulating metabolism, you're manipulating the electron transport chain. I think the fact that Ross holds steady does suggest that it is a very sensible way to change the electron transport chain. But I do think, again, looking at the liver papers and looking at the work previously published in manipulating the electron transport chain this way, we do have good backing to suggest that it is a safe way to look at ADPKD.
Okay. One more question is why do you have any idea why MCJ is increased in cystic kidney?
So given the data we've got, we did see an upregulation potentially in human data, but we haven't seen that mirrored in mice. So in general, I think that's work I'm still looking at is, in fact, MCJ increased, especially in the rodent models. If it was to be increased, I would be very much speculating here that it is a compensatory mechanism to disease. But again, like I said, it isn't shown to be upregulated in our mouse model.
1 minute.
And maybe one last question. Do you think that because lowering it is beneficial in kidney and liver diseases, do you think that targeting lowering this can also ameliorate liver cysts?
So we do actually, I think that would be something very interesting to look at. And like I said, we do have the liver histology and the liver samples. I do think considering whether or not this could be a treatment for PKD along with PLD would be very interesting, yes.
Okay. Well, thank you very much.
Thank you very much. Appreciate the questions.
I think that's all the questions. Yeah. Thank you.
I want to, again, thank the speakers for a wonderful morning session. We are now going to have a 14-minute break. So if we can get back here by around 11:30 Eastern time, we will start the next session. Thank you, guys.